My name is Randy Jirtle, and I live in Durham, North Carolina, and my phone number is 919-824-7151. And I used to be on the faculty at Duke University, and then when I retired, I became professor of epigenetics at North Carolina State University in Raleigh, and also adjunct professor at the University of Wisconsin-Madison.
And your educational background?
Yeah, my undergraduate, all of my education occurred, higher education occurred in University of Wisconsin-Madison. I got my undergraduate degree in nuclear engineering, and then I got my master's and my PhD in radiation biology.
At what point did you get interested in low-dose radiation and its effects on the human body?
Well, I've always been, because of my background in radiation biology, it's always, radiation effects has always been something that's been in the back of my mind, but it was not actively in it till way at the end. I was in the field of epigenetics, and we were the first to demonstrate that the fetal origins of adult disease susceptibility was due to alterations in the epigenome. And then we did a number of studies with other, this initial study involved use of nutritional supplements. But then we looked at other chemical agents and how if they potentially affected the epigenome too, and they did. So I then decided that I wanted to look at a physical agent. So that's then when I decided to look at the effects of low doses of ionizing radiation on the epigenome.
About what time was that?
The paper was published in 2013, so I guess this would be about 2011, something like that.
What brought you to this meeting?
Tony and the people that invited me, so Tony Brooks. We've been friends for many, many years. At my talk, I said that Tony was one of the sort of few people that was an assistant or associate professor at that time that, at least to me anyway, was more than willing to talk to graduate students, and I was a graduate student. And we just became good friends, and we even collaborated after that on something totally different from what we're talking about today. So I've known Tony for many, many years.
I have another question right on top of my tongue. I can't think what it is. What is your current feeling about the linear no threshold theory?
Well, again, I mean, initially I really didn't, I have to admit, relative to the people here who it seems to be something that's on their mind a lot, it was not really on my mind at all when we did these studies, because I actually thought that the effects of low doses of ionizing radiation would cause deleterious effects because of the linear no threshold model, which I learned when I was in graduate school, which clearly states that there is no dose that is safe. So we predicted then, based upon that, that we would see deleterious effects in this model system. We used the agouti mouse system, and you can sort of think about it as you have animals that go all the way from being completely yellow to brown, and that shift in coat color is due to alterations in a little transposable element that's upstream of the agouti gene. So it's epigenetically regulated, so it's a monitoring system for determining the effect of any kind of environmental exposure on the epigenome and therefore disease susceptibility. So our hypothesis was that in a dose-response manner, what we would see is increased numbers in a dose manner of yellow animals, which are obese, get diabetes, and get cancer at very high incidence. That's what we expected. The graduate student, again based upon the linear no threshold model, and the graduate student Autumn Bernal that did this work in my laboratory, we were going on with the experiments, they were continuing, and I never got any feedback from her. And finally, and I did never, but I finally said, what's going on with this experiment? Because we should be getting some data in. And her comment was, should I keep going?
Yeah, excuse me, we're having a film, we're filming in here.
Excuse me, can we?
Oh, I'm sorry.
It's all right.
You got to shut these doors.
Well, they're closed, but there's nobody out there minding them.
This happens once in a while, but not often.
So
just back up a couple of sentences and pick up.
So the graduate student that was doing these studies was Autumn Bernal, and I asked her then one day how this experiment was going based upon what we expected. And because I hadn't heard anything from her, and her comment was, it's really freaky. And I said to myself, that's not a good word to hear when you're in the middle of an experiment. And I said, Autumn, what's freaky? She said, we have no yellow offspring. All the animals are brown or heavily mottled, which means in this system that we now had observed with the effects of low dose rates or low doses of ionizing radiation, which was a single dose of 1 centigray or 1 rad or 1 rem, a positive adaptive response. And yeah, that's exactly what I thought. Wow. And my comment was even a little bit more severe than that. It wasn't even, oh darn. It was a little bit more than that. We are now in the middle of hormesis, which now means that we have to do a dose response rather than a single dose. And we're going to have potentially problems getting this published because this is a field that has a lot of controversy. So we continued doing it, and lo and behold, I mean, the paper was published in 2013. It was a clear example of hormetic responses. So it was exactly the opposite of what I thought, and it was due completely to alterations in the epigenome. So this is very important because in the past, not just here, but in cancer formation, all kinds of different biological responses, people have focused on the genome. So I like to think of the epigenome as being comparable to the software that's telling the computer, the hardware, the genome, when, where, and how to work. So in effect, a cell is a programmable computer. And that means now that you can alter with exposures to any kind of environmental factors, not only the hardware, which is usually mutations that you would see, for example, but also literally altering the programs. So you've got two systems really that are running, and in the past most people were totally focusing only on the hardware and not thinking at all about the software. So hormesis is due to changes not in the genome, and that's why you could never predict that this could ever happen. You would say there's no mechanism by which this can happen, because if you only think about radiation being able to alter the genome, it doesn't work. You should always have an effect no matter what dose you're at, and that's true. But when you're talking about the epigenome, now you're talking about software, and the cell has a biological response and can respond in both a positive or negative manner depending upon the dose at which you are exposed to. So ultimately you'll get a nadir, and then you'll start losing your biological effectiveness to respond to it, and then you get into what we always see at high doses of radiation. So as I said in my talk, hormesis is real, and it's due to alterations in the epigenome. But there are still a lot of things that need to be done, because now we know where to look. You don't look at the genome, you look at the genome, but you look at regulatory elements and then the effects that they have on expression of genes. So you get dose responses, all these kinds of things now need to be done, but we now have defined where to look. So the whole field has changed. Just like in the fetal origin of adult disease susceptibility, we have completely changed the way you have to look at this problem.
Outstanding. One other question. What would you like to see coming out of this meeting? What would you like the long-term effect of this meeting to be?
Well, obviously, the reason I came to this meeting is, one is that Tony invited me to come, but secondly is to provide this information to the people that were attending this meeting, because I was pretty sure that most of the people here would be more physics-oriented people and might not even know about the work that we had done, not just here, but all the work that we have been doing for the past 20 years in the field of epigenetics. So it was clear that this is an incredibly important contribution, actually, to the whole field of risk assessment. Because when you're only basing your information on epidemiology studies, you will always run into the problem that low doses that you can never really resolve clearly. Sometimes you'll see an effect, sometimes you won't. You know, it's all. But when you're talking about a mechanism by which this happens, now you can finally actually be able to understand it. I mean, it's not going to vary. It's always going to happen. This system probably evolved evolutionarily back at the one cell stage, when these organisms, these early organisms, were exposed to a lot of stress and a lot higher ionizing doses of ionizing radiation than we actually are exposed to. It also makes the point that in a way to optimize your health, we probably have to be stressed just a little bit. It doesn't necessarily mean that we have to be exposed to higher levels of ionizing radiation, because there are other ways that you can stress. Exercise stresses us. And the other thing we showed is that we can completely block this hormetic effect in this model system by feeding the mothers antioxidants and sucking up the reactive oxygen species. So it definitely involves the formation of reactive oxygen species. That seems to be the sensing system within the cell. How that's then linked to the machinery that lays the marks down onto the DNA and the histones that the DNA is wrapped around, that information has not been defined, and it's another incredibly important area of study that people are good in biochemistry, the ones that love signal transduction, they're going to love this because it's a whole new field to look at. I wish I was young again, because I would just love to get back into doing these kinds of studies because they're so exciting. And we're going to understand this. I don't think it's going to take as long as we think, because with the sequencing techniques and stuff that we have now relative to what we even had 10 or 15 years ago, these things can happen quickly now, and in the past that would have taken a long time. You just have to know where to look. And we now know we have to look at epigenetic changes and changes in expression of genes, not mutations.
If this conference results in one recommendation to the general public, what would it be?
I mean, again, I'm not a pro in this area, but, and as I said, I wasn't even I didn't even think hormesis existed before we started this study, is that low doses of radiation are not as harmful as people think. And if anything, they might even provide a stress system that actually optimizes our health. It's not, I wouldn't say necessarily go out and necessarily get irradiated, but you need some stress in order to optimize your health. And radiation is one way in which that happens, just naturally. Yeah.
That's a cool sentence. You got anything?
Yeah, I do. And I'm not an expert, so I just go right to the shiny object. Tell Jerry, you said, well, this is controversial, so people aren't going to really, this may or may not be accepted. I mean, you're a scientist, you're doing best practices here. Why wouldn't people? I mean, why wouldn't people believe the science? What could possibly be controversial? How is this controversial? Please tell Jerry.
Well, the reason that this is controversial, because the field of radiation biology, or the first time people were introduced to the biological effects of radiation were with Hiroshima and Nagasaki. So, I mean, with a massive number of people being killed from the bomb and some from late damage. So, and then we have a linear no threshold model, which basically says there's no dose that's safe. And now, with what information we have now, it's clear that that's not correct. But it's going to take time to educate people and tell them. The best way to educate people is to provide mechanistic methods and reasons for why this occurs, not epidemiology, actually. That's important. But ultimately, you have to have a mechanism for people to take these things seriously. And we now know the mechanism. It's epigenetic. We have to do a lot now to define the dose responses and dose rates and all these type of signal transduction pathways that lead from reactive oxygen species increases within the cell and laying down to these epigenetic marks. All that has to be done. Which genes are even being altered? We don't know that either. But now we know where to look. And that's what needs to be done in the future.
Perfect. You're going to have to go back and restart your camera. It's almost 10:30.
Okay, I did have one follow-up, but I've got Joe Joe is gonna. I told him how to press the button so he could start. I just want to get one last thing. So are you saying basically, and restate this for me, that LNT makes sense if in a vacuum it makes sense, but you're not the low dose. The reason the low dose is controversial, you think you've kind of found that, and that's this epigenetic piece that kind of mitigates the low dose piece. Is that, am I understanding that correctly?
Yes, because
please explain that just very succinctly, like a 2-sentence version.
Well, radiation, when let me start over. When a cell or a body is irradiated in the cell, it's affecting not only the genome and causing potential mutations, but it's also the other thing that's exposed basically are the programs that are running in there. And that, so there's two systems that can be altered. If you focus only on the genetic information, the linear no threshold model would be correct, but we don't have just that. We have a programmable computer. We don't have a hardwired computer. We have a programmable computer, which means now that you can affect not only the hardware, but also the software. And it's the changing of the software within our cell with the exposure and the generation of reactive oxygen species that allows the biological system to, in effect, give rise to a positive adaptive response. And that's what you see with the phenomena of hormesis.